Hello friends, a warm welcome from Pegasus Agriculture Academy, a free online coaching platform for agriculture related exams. Okay, so here we are dealing with the subject group Agriculture Microbiology, which comes under the major subject group Plant Sciences. Okay, and the previous chapter we saw about the introduction to microbiology, and in this chapter, let me explain you some information about the history of microbiology. Since this part is very long, it has been segregated into two parts, that is the part 1 and the part 2. Without wasting any time, we will go into the topic. And friends, our previous video I have told you about what is microbiology, what are the different subject groups, or what are the informations that recorded needed for the introduction classes. Those who have not seen the video, please see the video that is present in our playlist, or you can click the I button to get the link. Okay, the link is also provided in the description. You can check it out. Okay, so what are microbes? Microbes are organisms that are very small that cannot be seen by our naked eye. Okay, so the organisms that are very small, they are about 1 millimeter, or they are very less than 1 millimeter in size. Okay, so in order to see these microorganisms, we need a special apparatus called it may be a microscope or it may be a very good magnifying lens, but we need some aid to see these kind of microorganisms. Okay, so the discovery of these microorganisms or the discovery of microbial world started with the discovery of microscope. Okay, the discovery of microscope by Anton van Leeuwenhoek. Anton van Leeuwenhoek is just a cloth merchant. Okay, he is not a scientist. He is just a cloth merchant. And in previous year, the cloth merchants they used to have some kind of magnifying lens so that they can see the quality of the cloth and also the texture and the fineness of the cloth. So one such person is Anton van Leeuwenhoek. He prepared his own lenses. Okay, so in the year 1674, Anton van Leeuwenhoek he discovered a crude form of a microscope. Okay, so this was the microscope invented by Leeuwenhoek. It consists of a simple handle. Okay, and then it consists of a focus knob. You can see, you can follow my mouse cursor. Okay, so it consists of a focus knob and it consists of a lens that is attached to a brass plate, and behind the lens there is a sample holder. Okay, so you can see the sample, you can keep the sample in this holder and afterwards you can view through the lens. Okay, and what happened is he used Anton van Leeuwenhoek. He saw very tiny structures and that used to be even living structures he viewed through this crude type of a microscope. Okay, and after seeing this he started writing letters to the Royal Society of London, and in his letters he mentioned these organisms as animal cubes. Okay, so remember this, it is very important. The organisms, that is the tiny microscopic organisms, were called as animal cubes by Anton van Leeuwenhoek. Okay, and his microscope is capable of magnifying the objects up to 300 times. The magnifying object up to 300. So if an organism is about 1 millimeter, when you see these microscope, it will be magnified to about 300 times. Okay. So remember this, Anton van Leeuwenhoek is not the person who invented the microscope. He is the person who first used a special kind of microscope, or he used to develop a very advanced microscope so that he can see microorganisms. Okay, so before Leeuwenhoek, there are certain persons who used a microscope. Okay, before Leeuwenhoek, during the year 1590, there is a father-son duo called as the Hans and Zacharias Johnson. They were the first person to use such kind of microscope. Some studies indicate that they created the first microscope. But what happened is, during the time, there was different persons who used to discover different things of their own. Okay, so you don't have contact with anyone, so at the same time different persons they will be using different kind of microscope, so there is a no clear cut history who discovered the microscope, but most of the studies indicate that these two persons, that is Hans and Zacharias Johnson, they discovered the microscope. Okay, but one clear cut thing about this is they used or they invented the first compound microscope. It is a very confirmed study. Okay, so compound microscope is nothing but the microscope that we use in our lab. Okay, it consists of one objective lens and one ocular lens. So what happens is, if you use two lenses instead of one, the magnifying capacity will be increased to a very maximum height. Okay, And the magnifying capacity of the microscope that is used by this duo, the father son duo, is about 50 to 100 times. Okay, and afterwards, a few decades later, the another person, another important scientist is Robert Hooke. Okay, and he also invented a microscope that is capable of magnifying organisms or magnifying structures to a very bigger size. Okay, he wrote a book called as the Microphagia, which outlines about the various studies using his microscope. And he is the first person to formulate the basis of cell theory. And Robert Hooke is the person who coined the term cell. Okay, and the magnifying capacity of the microscope that is used by Robert Hooke is 200 times. Okay, so the magnifying capacity of the microscope that is used by Leeuwenhook is 50 to 300 times. At the same time, the magnifying capacity of the lens, the microscope used by the Jansens is 50 to 100 times, and Robert Hooke it's about 200 times. Okay, so you can see that the magnifying capacity of Leeuwenhoek microscope is higher than these two, and that is the reason he is capable of seeing microorganisms so clearly. Okay, and that is the reason he is called as the father of microbiology. Okay, so in the history of microbiology, the first thing that was discussed in the previous year or in the olden time is the spontaneous generation theory or the theory of abiogenesis. Okay, the spontaneous generation theory is nothing but the previous people or the older people or the older generations, they used to think that living matter, they arise from non-living matter. Okay, even there are certain persons who supported this theory. So the first person to support this kind of theory, or we have a written establishment about the supporters, is Aristotle. Okay, the famous Aristotle. What is said that non-living matter, they consist of a vital heat which produces, which is capable of creating living matter from it. Okay, and he also showed some evidence. He said that the sudden appearance of fish near new pool. Okay, so we will have seen in case of some lakes or in ponds, during the dry season there will be no water and no fish will be available. But suddenly after the rain or during the rainy season, the water in the pond or the pool, they will get filled up and you can see fishes. Okay, so they don't know that the fishes are brought by means of the rainwater from some other places, or the eggs that are situated in the that are present inside the pond during the dry season, they may get hatched out during the new rain. Okay, so the first person to support the spontaneous generation theory is Aristotle. Okay, and next person to say, support this spontaneous generation theory is Hellman. Okay, what he said is, he said mice and other maggots that arise spontaneously from the non-living matter. Okay, so he also did an experiment showing that he kept some rags and wheat kernels in the open space, and after few weeks, when he opened these rags, the mice started running out. Okay, so he said that these mice, they arose from the non-living matter. Okay, likewise he kept some meat material in the jar, and after a few weeks, he used to be, he said that there are maggots and other flies that are present inside the jar that is present on the meat, is they arise spontaneously. Okay, and the next person who suggested this theory is John Needham. Okay, and remember this. There may be a question in your exam. Who are the persons, or the persons who supported spontaneous generation theory? They may give the three scientists' name, and afterwards another person who does not support. Okay, so they may ask who is not a supporter of abiogenesis theory. So you have to memorize the supporters of the spontaneous generation theory. And the third one is the John Needham. What he did is, he used or he took a broth, he kept the broth, or he boiled the broth in a flask, and afterwards he sealed the flask, and he said that microbes arise after a few days, so they do not come from the outer side. He said the maggots, they were already produced, or they are produced by means of spontaneous generation theory, or they arise from the meat itself. Okay, but he forgot one thing: the boiling used by John Needham is not enough to kill all the microorganisms that is present inside it. Okay, so the ordinary heating, they won't kill the spores or the heat resistant microorganisms. They are not killed by means of John Needham's experiment. Okay, so these are the supporters of the spontaneous generation theory or the theory of abiogenesis. Okay, and then came the persons who started disproving this theory. Okay, so the first person who started disproving the theory of abiogenesis is Francisco Redi. Okay, remember this, this question was asked in most of the examinations. Okay, the first person to disprove the spontaneous generation theory is Francisco Redi. Okay, so what he did is he proved that the maggots did not arise spontaneously from the rotten meat. Okay, so we have seen that the supporters, they said that the maggot, they started appearing from the meat spontaneously. So first thing Francisco Redi started doing is he wanted to disprove this theory. Okay, so what he did is he took a glass jar, he kept the meat inside it, and he left it open. Okay, after a few days you can see the flies and maggot, they started growing on the meat. Okay, and then he did another experiment where he took a open jar, glass jar, he put the meat in it and sealed it with the cork. Okay, so after few days you can see no maggots or any flies are seen started growing in the closed structure. Okay, and then he took another flask, he kept the meat inside and closed the flask by means of a wire mesh. Okay, and this wire mesh, on the top of it, the meat and the maggots and other flies, they started growing in it. Okay, so he proved that the maggots and the flies, they are not present in the meat, they came from the outer surface. Okay, or the outer environment. But some say that what happened is, even in the sealed flask, the meat started rotting. Okay, so when we see this meat in a microscope, they started seeing the presence or the rise of microorganisms. Okay, so he failed to prove that the appearance of microscope. So still the persons who supported the spontaneous generation, they don't accept Reddy's experiments. Okay, and next came the another group of scientists who started disproving this theory. Okay, so first one for doing it, the Lazarus Spallanzani, and discovered a technique called the hermetic sealing, and this is the foremost for other packed foods and everything. Okay, he is the forerunner of the discovery of packed foods. So what he did is he used a technique called the hermetic sealing, and what he did is he took a bottle. Okay, he kept the beef broth inside it. He closed the broth tightly or the flask tightly, and then he heated the flask for a higher temperature, and so that even the resistant structures, they cannot survive. Okay, so he proved that the microorganisms, when heated properly, they won't arise from the meat. But due to his bad luck, at the same time oxygen was discovered. Okay, oxygen was discovered, and it was confirmed that they are the life forms for living organisms. So what the supporters of spontaneous generation theory said that the experiment lack oxygen. Okay, because he sealed the flask completely so that no oxygen can enter into the flask. So Lazarus Spallanzani experiment cannot prove, completely cannot disprove spontaneous generation theory. Okay, and next the person is the Franz Poulet. What he did is the same experiment. He took a jar, he made a tube of the jar. He closed the jar by means of a tube, and he passed acid through the tube. He passed the air through the tube after passing through a strong acid. Okay, so that the air, before it get contact with the broth, it was completely sterilized by means of the acid. Okay, and then another experiment done by Swan is passing the air through a red hot tube. Okay, so next Vandus, what he did is he used a cotton plug to filter the air, so that instead of using the strong acid or red hot tube, he closed the cotton plug mouth. Okay, the with the cotton, and so that the air that enters into the mouth of the flask is very completely sterilized, so that there is no growth of microorganisms. So what happens is, whenever they discover or started disproving the theory, they suggested the supporters of spontaneous generation theory, they suggested a new thing. Okay, so what they said is after passing through a strong acid, the chemicals or the strong acid, the life forming material that is present in oxygen is killed. So that the reason there is no growth of microorganisms. So, and friends, if I am going fast or if I am moving uh, the slides very fastly, you can just pause the slide, so and we can make notes of it. Okay. And then came Louis Pasteur. What he did is he is the man, or he is the hero of microbiology. Okay, this is the first time Louis Pasteur came into the limelight. So what he did is he disproved the spontaneous generation theory once for all. Nobody can say any um, bad suggestions, or nobody can challenge him after he disproved this spontaneous generation theory. And the experiment that was performed by Louis Pasteur is the Swan neck flask experiment. Okay, so Louis Pasteur is not a microbiologist; he is a French organic chemist. Okay, who demonstrated a lot of experiments to disprove the spontaneous generation theory. Okay, so what he did is he took a flask. Okay, and the flask neck he designed it in a way like a neck of a swan, okay, or a S-shaped tube, okay. Then he heated the liquid so that the air that is present in the liquid gets out of the previous air that is already present inside the flask. They are flushed out, okay. Afterwards, he made allowed the liquid to cool slowly. So what happened is the dust and other microorganisms that are present in the environment, they get settled in the S bend, okay. So only the air without the microorganisms they enter through the flask, and even after keeping it for a long time, the broth will remain very sterile. Okay? So people cannot say that the life form or the vital forms that are present in the oxygen is destroyed by means of any experiment. Okay, before the experiments by Scuders and experiment by Scavan, they used gun cotton, or they used they used acid, or they used heat in order to sterilize the air. But Louis Pasteur experiment, he didn't do anything. He just made a flask so that the dust and other microorganisms they get trapped into the flask, okay, trapped into the husband. And then after a short time, if you bend the liquid or bend the flask so that the medium or the broth that is present inside the flask, when comes in contact with the S band, it will get contaminated, and afterwards there will be a growth of microorganisms. And this swan neck flask experiment uh, completely destroyed the belief of the supporters of spontaneous generation theory. Okay, so the next person in the process of disproving the spontaneous generation theory is John Tyndall. Okay, so what he did is he confirmed all the suggestions or all the informations given by Louis Pasteur as 100% correct. Okay, so he proved that dust carries the germs which are responsible for contaminating the broth. Okay, so dust carries the germs and other bacteria, other spores and everything, so that when the air from outer surface they enter into the flask, they make the broth contaminated. Okay, he developed a technique called as the Tyndallization. The Tyndallization, it's also called as the intermittent heating, or intermittent heating technique. Okay, so Tyndallization is nothing but a process in which we will heat the or sterilize the broth continuously or consecutively for 2 or 3 days. Okay, so what will happen is if you heat the broth for the first time, the ordinary structure, the vegetative cells and everything, they will get restored. At the same time, the heat resistant spores that are present in the broth, they will start sprouting. So you will heat the broth on the next day. Again, the sprouted or germinated spores, they will also be killed out. So if you do this experiment or this do this sterilization for 3 consecutive days, what will happen is the broth or the material that we want to sterilize, it will be completely devoid of any microorganisms. Okay, so these are for the part 1 of history of microbiology. So in the previous video I asked a question. We will see about it. We will go into the question section. Okay, so in the previous video I asked a question: where is National Bureau of Agriculture Important Microorganism is located? The answer is Mau, okay, or the Maunat Bhajan. So this NBIM is located at Mount Bhajan, Uttar Pradesh, India, and it was started by Government of India in the year 2001. The main aim for starting this institute is collection, maintenance, and preservation of agriculturally important or beneficial microorganisms. Okay. For today's section, we will see some previous year questions. That is a memory-based question. The first question is, who coined the term cell? You can give the answer in the comment section. Yeah, you are right. The person who coined the term cell is Robert Hooke. Okay, and one more question. Anton van Leeuwenhoek, in his letters to the Royal Society of London, called the tiny organisms as. We have seen this in the start of the video. Those who know the answer, you can give the answer in the comment section. Otherwise, I will discuss this in the next video. And friends, as I promised last video, I said that we'll be giving the books that we have to follow for this ICAR JRF preparation. The book is if you have a very short time, okay, like few months, 1 or 2 months or something, you can use this book. You can follow this book, Introduction to Microbiology by Taran Kapoor. But this is an old edition. It may be available in your college library, or it may be have some difficulty to do this one. Okay, and if you have a very long time, that if you are in the third year or if you are in the start of the fourth year, you can follow this book, Introduction to Microbiology, and this is the advanced book or a little bit added of this previous book. Okay, so if you have a very long time, you can follow this book, and if you are very short of time like 20 or 30 days, you can follow your UG notes. Okay, we have two courses like introduction microbiology, principle to microbiology in the first year, and then in the third year you may be having applied microbiology or soil microbiology. You can follow these notes if you don't have any time, but if you have some time, you can follow these books for our further studies. Okay, and friends, you should all at least have a material, or you should follow some book, because the information that I am providing in my video will be very short, and when you see this or when you read this in a book, you will get a clear picture of it. Okay, and best of luck for your studies. Thank you.